All right, thank you. Welcome, everybody. Thank you for coming. It's a great pleasure to have here today Professor Jelena Kovačević from at CMU. Dr. Kovačević got her PhD degree from Columbia University, and following that, she joined the Bell Labs for a number of years. And then in 2003, she joined the BME at first. BME department at Carnegie Mellon in Pittsburgh. And now she's actually in the ECE department, the current department head, where she holds the Edward David Schramm professorship in the department. She's also affiliated as a professor in BME. She got several awards, one of them being the Dodd Fellowship at CMU, Belgrade October Prize, and the EI Jury Award at Columbia University. She's been co-author and author of many award-winning papers, and also a very well-known author of a very well-known book on wavelets, one of the first books published on wavelets and subband coding, and more recently a very good book which I highly recommend. It is called The Foundation on Signal Processing. So for those of you who actually are into signal processing in a very serious way, you should buy it.
Thank you.
There you go. And
it's not like I'm going to get rich off it anyhow. So.
It's the worst way of getting rich.
Yeah, exactly. Yeah. You don't.
Dr. Kovačević is a fellow of IEEE, and she was the editor-in-chief of the Transaction on Image Processing, and she's been a keynote speaker at several conferences and meetings. And her research interests lie within multi-resolution techniques, graphs, biomedical imaging, and smart infrastructure. And it's a pleasure to have her here today to talk to us about some of her graphical signal processing. And thank you for coming. The floor is yours.
Thank you very much. I'm really happy to be here. Hamir has been trying to get me to come here for a long time, and I apologize for not doing it earlier. And then we have Dan Stansel, who was at CMU for a number of years, and he's your current department head, so we see each other in that capacity as well. So as a current department head, I will apologize in advance, and you can sleep through those 10 minutes, but I feel it's my duty to tell you something about CMU and our EC department at CMU. And if you find any good ideas, feel free to steal them. And I actually, so funny, yesterday, just as I was leaving, I got mail and I got your newsletter in the mail. So I was reading this on the plane, so I got lots of good ideas that I'm going to try to steal from you. So, okay, I'm going to skip my picture. I just want to thank all our faculty, staff, students. I couldn't put all their names in a word cloud because there are 1,200 of them, alums, and just to tell you a little bit about CMU, this picture on the bottom is Arthur Hammerschlag and Andrew Carnegie in 1908 when Carnegie Tech was actually formed. This is the famous, this doesn't really show on the screen like on ours. Yeah, this is the Hammerschlag whole cupola where we sit, and this is the hallmark of Carnegie Mellon. You see it there again. And I've been walking around this campus extremely jealous of the beautiful space you have and large classrooms, and and what happens on Carnegie Mellon campus. It's a tiny campus. We have very little space. So when they build something, you can see the latest building is caught hole energy. You see that it's on stilts. So you find a little tiny hole anywhere on a hill, you build a building because there's no other way to get the space. So here's Hammerschlag. This is, for those of you who know, this is the CIC building. And that was just like a little hill here, there. And they built the entire building on this. It's pretty cool. Anyhow. Just to give you a little bit about our department numbers, we are about 1,400 people altogether. That's 70-something faculty who sit also in different locations. Not all of them are in Pittsburgh. We have about 80-plus staff and another 1,200 students, graduate and undergraduate students. As I mentioned, we say sun never sets on our ECE because we have programs at different locations. We have another location in Silicon Valley. We have 11 faculty members sitting there in about 180 students. We have a dual degree program with Portugal. We have a dual degree program, PhD and master's with China. We have a master's program in Rwanda, so it's pretty exciting and a challenge for me. So we are pretty hardly ranked. The red lines are computer engineering and the white lines are electrical engineering, though we give one degree. We give an ECE degree to our undergrads. We're still ranked separately by these. Computer engineering is consistently ranked in the top 5 and electrical engineering somewhere between 5 and 10. And the college ranking have been also steadily rising over the years. We're doing pretty well there too. And one that we should be proud of is that they voted us in one of these by the STEMinist thing as the #1 STEMinist school. So that's cool. Getting more girls, it's more fun for everybody, right? We have about $50 million plus of annual budget. Our enrollment has been going up steadily. It's mostly due to our master's program, like I guess everywhere else. We keep our bachelor's numbers pretty steady, and that means that the selectivity has gone down, so now we are somewhere around 12–13%. These are the numbers of our graduates from last sort of academic year. This is what our undergrads like to look at and tell their parents. If you look at the average salary for bachelors is like close to $96,000, which I think is pretty insane, but they're excited about this. And, when they leave, they do get, they're roughly, I guess, you can look at it over the years, half to industry and grad school, and there are very few who don't have the job once they graduate. We have spent a couple of years in strategic planning. Why is that important? Because it was important for us to put on pen onto paper and figure out where we are going. We have specified what the core values are, and believe it or not, even though people think like, okay, this is really not important, we actually fought really hard about every one of these words. And what we see where we want to go, so vision is sort of like the North Star, where you want to end up. It's kind of an aspirational goal. How you do this, the mission, and then you have the strategic objectives, which we called our FIRE, strategic plan. And I played with this because we had these 4 core areas, and there is a place on the web where you can put actually letters, and it gives you all possible words that you can make out of it. And I thought FIRE was really great, but as soon as I showed it to faculty, they said, where's the D at the end of it, right? Okay, so, there are really no particular order except to make out a nice word. They're about fostering an environment, which is really important for us. Carnegie Mellon has a very inclusive and particular culture that we're sort of guarding with our lives to impact society. That is another thing that's very important to us, that our work and our students go out there and have a measurable impact on the world around us. And then, of course, research and education. And we have just released this plan. So let me just tell you, a couple of things about some of these. We have a number of student organizations. I don't know how it is here. I would love students to be even more involved in them because they help them, develop a better sort of environment while they're at CMU. But some of the things that we have done, we started giving them de-stressing kits around quals, around tough classes, midterms, and so on. These are little baggies with chocolates and things inside. We have started to address the issue of what people call a sophomore slump. For the undergrads, it seems that the sophomore years is very, very hard. And one of the big predictors of them being successful and going through this successfully is having more contact with us, the faculty. So we started doing things like social contact, not only in classes. So we would take them, let's say, to the Cathedral of Learning, which you know what it is, but it's a cathedral at the University of Pittsburgh campus that has classrooms from all over the world. I had like a puzzle night, so you see the puzzle with the cupcakes there with undergrads. We had their goals for the year, and they put ECE, they put little post-it notes, and these are some of the just keep swimming. It's kind of good for all of us. We have increased the number of undergraduate female population. This was in 2006, somewhere around close to 15%. Last year, we were close to 26%, and this year we're going to be even better. So I'm very proud that we are able to attract more girls to come into ECE. And one of the reasons I think for the low enrollment is that in general, and you can agree with me or not, but I'm curious to see if anybody had an opinion, was in high school, lots of kids will be exposed to some form of computer science, but very few are exposed to what electrical and computer engineers do. There are no classes, and they don't even have an idea as what we do. So I actually did something where I created this little YouTube video on what do electrical and computer engineers do on an example of an iPhone, because I wanted to disassemble an iPhone and show them everything about that phone is really what we created, from the touch screen to the wireless to the coders to apps and so on, operating systems. So it's really important to and this is up to you. I'm assuming these are mostly grad students here, right? It's up to you also to show, to tell the kids, look, here are the cool things we can do. They don't even know what kind of work comes out of our profession. And clearly, the great weather in Pittsburgh goes a long way towards creating a wonderful environment, the outdoors. I stole these slides from my predecessor. These are really, I love the great slide. Look at this. The Badaki came to Pittsburgh. The local culture, does anybody know what that is? Terrible towel. You guys are not into, except for you, right? It's a football thing, and I'm not a big football fan, but okay. And just in terms of impact, you can measure it any way, you want, but there are several of them you can do. This is our Professor Anthony Rowe, who was at the last World Economic Forum in Davos. This is the new Dean of Computer Science, Andrew Moore, who was in Google. So there's this is all CMU, these are all CMU faculty, and it was a large delegation that participated there, and I think that's very important for us, for all of us actually, to show what our discipline can do. We have started a larger communication effort. We didn't do anything on this. CMU was kind of rather, I would say, amateurish when it came to selling our work. So we're trying to let people, and selling in a good sense, letting people know what we do. From creating the electronic version of the newsletter to the printed one, to putting our strategic plan on paper so that people understand what our thrust areas are. And, you can see that, I think, in puzzles a lot. So these are like theoretical and technological foundations. These are application domains, and these are systems level things. And, I sort of think that we as ECE have, an opportunity to distinguish ourselves by saying we are system builders, because we can do things from the physical to the cyber domain that nobody else can do, right? And these are, creating these position papers for each one of these puzzle pieces, where, again, explaining to people and to kids and the prospective students what we do. So, each one of these in front has something that they understand sort of in layman's terms what, for example, data and network science is, and on the other side is a more specific, like, projects that we do. And, going on social media, creating faculty research videos. As you all know, most of the time when you want to learn about something, you go and watch, if there is a little YouTube video, that's very useful. So we did a lot on recording what we do, like 3 to 5 minute videos. And they are really very, very useful. And that's the one that I told you about, a little YouTube video about what's ECE. I encourage you to go see it and see if you agree with this or not. And then, you can do it in the number of patents we have, the companies that our faculty spin, the alums in leadership positions, where our students end up in terms of industry, how to mentor them. So we have created this online mentoring system now where the alums can sign up and say what they want to mentor students in. Let's say they're willing to proofread the resume, and the students sign up and they can get matched. So this is a new thing we are doing. And then in research, just a couple of things I wanted to say is that we are ECE, and it's very easy to think about this as EE and CE. We never think about it this way. So we don't think about it as two sides of the umbrella. We think of it as in one big area, which is really dominated. These keywords that you see here are really taken from the faculty keywords of their research. And so you can see what comes out the biggest, right? Systems that appears most often. It makes sense, right? Because this is what we do. And again, I told you about the thrust areas. We are expanding, so that Scott Hall that I showed you before kind of sits in here, and we're going to have a new 12,000 square foot nanofabrication facility, which is going to help us, sort of become one of the top, facilities in the country. So far, what we have is way sort of outdated and obsolete. And again, these are numbers in research expenditures. This is a self-driving car that Professor Raj Kumar has been working on for years. Has driven actually 30 miles between, Pittsburgh, going to the airport and back. I think this is somebody from the governor's office who's sitting on the other side of the car. He was brave enough to sit in it. And finally, in education, I think we characterize ourselves by the maker culture. Build 18 is an effort that a group of students started a bunch of years ago, where they said, we want to do something fun and build fun things, but we don't want it to be a class. So they self-organized and they work on it the entire fall, and then the first week of classes, they demo it, and they have industry support. It has become incredible. It has become something now that's university-wide. They have over 300 people build cool things. And they come up with what they want to build. So I saw one where they build a Harry Potter chessboard, which is, remotely operated, and they play Harry Potter music. It's really great. It's a great way of engaging undergrads. But they did it themselves. They also raise money from companies for sponsorship and so on. We think that our students can do anything, and this is usually characterized, I think, if they can do math, they can do anything except play football. This is a game, I think, in 1948 that they lost 28-0. So you can see there's lots of football references in anything that has to do with CMU. I would like to see our students become broader in terms of interest so that they can expand their minds at the time, in their lives, especially undergraduate students. So this picture here is because we had a student who just won the Martin Luther King poetry contest. So I kind of like to see ECE students doing something outside of ECE. And finally, as I said, we are global, so we have a number of classes. So this is from Pittsburgh that are beamed to Rwanda, Portugal, China, and Silicon Valley. These are the number of classes from Silicon Valley that are beamed to other locations, and we had 2 classes from Rwanda. So we are developing also this portfolio of either online or basically broadcast classes between locations. And that's it. I don't know if I doubt anybody has any questions about this, but if you do, my job is done. Now I can do science. Okay? So let me go to the other one. Sorry, this is a weird thing. For some reason, PowerPoint wants me to put it in a different window. So let me go to this one. Okay. So I imagine that this is your standard seminar that people come from various areas. So this is a rather technically shallow talk. And it's more a story of how I went from biomedical imaging to graph signal processing. I don't think it's an obvious thing. And I kind of got amused myself one day thinking about how did we get there, connecting the dots. And there are lots of interesting things that happen along the way. Might be interesting for you or not, I don't know. So this is just to thank people who are in my lab currently. It's not a very big group anymore, given that I do what I do, so I had to kind of shrink it a little bit. These are previous alums and my collaborators, more senior ones. So let me talk about this a little bit. First of all, it's not an exhaustive talk, it's really a story, and it is in some way chronological, and of course it's not unbiased, it's totally biased from my point of view. So all of this started with me looking into bioimaging problems, and to explain it to myself and to organize it in my head, I kind of created this sort of block diagram of how I see a bioimaging pipeline. So you can imagine here that you have some sort of a specimen, right, that you're taking, let's say, on a microscope or some other device. And the PSF means a point spread function. It's basically a filter that models that acquisition device, because that specimen or something needs to be acquired, right? It could be a biopsy, it could be, fruit fly, it could be all kinds of things. And then once you acquire it, there is A to D conversion that happens, right? You have to digitize it to be able to get an image that you can process and that you can operate on. And then these things here, because there are lots of arrows, you can see that you can skip any of these processes. When biologists or doctors look at this pipeline, I just try to put the appropriate things in the appropriate place. That doesn't mean you have to go through all of them. So for example, if I wanted to invert the effects of the filter and this A to D converter, which introduces noise in the process, I could first invert this one by denoising and then the other one by deconvolution. This means, reversing the effect of the filter here. I don't have to do this. I can skip it. Sometimes people do it, sometimes they don't. Or I can do what people in imaging call restoration, which is these two things together. I was just trying to see where the imaging, the standard image processing tasks would fit in this picture. And then another step that you might have to do is when you acquire images, let's say that you acquire something that's a 3D volume. It's on a stage, and this stage can jitter. It's a mechanical device, right? So as you acquire images, it could be that you lose the reference point along, let's say, the z-axis. So once you acquire them, you need to be able to realign them, which is really registration. You could also do, which biologists do a lot, they would change the magnification of the microscope and take things with different magnification to see more detail or to see less detail. When they see less detail, they can take a bigger field of view, right? When they want more detail, they have a smaller field of view, which means sometimes they take a bunch of small, like high-resolution images, and they have to stitch them together to get a big image, which is what we call mosaicing. So this step could exist. And then the next step that very often happens, or most often happens, is some sort of segmentation, tracing, tracking, because you're trying to count objects or, take the object away from the background or trace something, let's say, on an axon or track something in time. These are all sort of segmentation type tasks. And the final box is sort of a catch-all where you do some analysis and modeling. And this is where any sort of data mining happens, where you're trying to extract the knowledge. And this knowledge is typically guided by some sort of a hypothesis of your collaborator. They have some sort of an application they're trying to figure out, okay? And what happened really in the last 25 years or so is that an explosion happened in biology with the advent of new acquisition devices, especially in fluorescence microscopy, where they discovered a fluorescent protein in a jellyfish. The first one was the GFP, a green fluorescent protein. They actually got a Nobel Prize for it. And what they were able to do is, in some way, if you want to think about it, either attach that protein to something of interest in a cell and then, take an image of this by using, you shine a laser light, it excites it, the energy of the electrons changes, they change, orbits, they release the photons, you catch the photons, you record them, and that's your image, right? Or sometimes they're able to code it into DNA, which to me sounds like science fiction, and basically the new DNA will express this green fluorescent protein, and then you can also take an image. And what's amazing is that you can do some of these things in live cells. So you don't have to kill cells to do that. And not only do you have now green fluorescent proteins, but you have fluorescent proteins of many different colors. So you can actually mark things in the cell, take an image, and see what's happening. The progress of a disease or cell tracking and so on. And so what I'll try to do here is, when I was looking at this, it was fairly ad hoc what people were doing in biology and medicine, because most of the time they would take something, they will take the first tool that comes to mind, whether the tool was really applicable or not, and they didn't quite understand what this tool did, so they couldn't really figure out what the appropriate tool was for the appropriate problem. And so the way we think about it is sort of, you have to have some set of guiding principles, that will inform either how you're going to create your algorithm or a mathematical framework or both, but it's kind of an iterative process. And the way I think of it is really this way. Typically what I was inspired by was some application that I have gotten from a collaborator, and then I would design some sort of an algorithm or a mathematical framework, but typically I would try to do design an algorithm based on what I was seeing that needed to be done, And typically I would find out some results that would make me think, okay, can I put a more formal framework under this so that I don't have to ad hocly do this every time I have a similar application. And so you can do in one way or the other way, or you can, kind of oscillate in between. So this is going to be the guiding picture for the rest of the talk. So to show you some of the characteristics of these images, depending on what they are, this, for example, is a fluorescence microscope image. It's very, very weak. It's like a weak biosignal. It's very, there's not much that you can see that. So you can have low contrast and poor definition, lack of consistent edges, which is something that people in image processing really like to use edges for processing. If you don't have them, creates a challenge. In particular for fluorescence microscope image, you have this punctate appearance that looks like sort of almost like a drawn from some sort of a statistical process. You can have acquisition device artifacts where you're not sure whether the fact that this is dim is because of the acquisition device or this is a real biological or physiological change that we are seeing in this image. You have all kinds of noise. As people are shifting towards non-invasive procedure, especially in medicine, you are putting, you are recording things farther and farther from the source, right? You can, open somebody up, take a biopsy, take the tissue sample out, and, they still do this. But whenever possible, people, the doctors don't like to do that. So there are all kinds of noise, electrical noise, neuronal noise in our brains, mechanical noise. So modeling of noise is another issue. So what I'm saying is I'm just showing you how this differs from the typical images that we dealt in image processing for years, which are mostly natural images, right? Images that you take with your camera around here or outside. You have humans and animals as customers, meaning things are now, there is a wide range of what's considered normal. So it's really hard to pinpoint. These are not discrete points. These are ranges now. There are large deviations. For example, what's normal for me when I take a blood test might not be normal for you. But, there is still a range of what's considered normal for a population. Could be human population or population. These are fruit flies here, fruit fly embryos. And another big problem is that very often we don't have access to the ground truth. So you deal with almost ill-posed problems. You're trying to create algorithms that are going to train towards something that could be inherently wrong to start with. And this is the part where I think, I started thinking using what I have learned before and what my expertise was. When I look at this image, what I see first is this big blob, right? The big blob is sort of like a global feature. But if you come closer to it, you have the little tiny bluish, purplish dots. These are nuclei. So you have also fine features. So this is, in terms of an image, a non-stationary object. And that made me think that perhaps multi-resolution as a tool could be useful in images like that. And we use them. So some of the principles that we have followed, and to be honest, I think some of them are rather intuitive as we were going along. And then when I looked back, I tried to put on paper what are those principles that I believe we followed. And some of them you could learn from the problem. So you could learn from the data, use as much a priori information as you can. And sometimes, if you can, mimic the human. That's a hard thing to do. Also, we used a lot of multi-scale and multi-rate tools because we can look at local and global features. We ask ourselves very often, what's the trade-off between universality and specificity? And what I mean by this is you could take an algorithm that's off-the-shelf algorithm, try to apply it to the problem, and that's perfectly fine. But to really make it work for that problem, you will need to make it more specific and more adapted to the problem. So the question is, when you're developing something, if this is a one-time use and it's not going to be widely used, is it really worth spending 4 years on developing something that's very, very specific to that problem? Probably not. But on the other hand, if you're developing something that's going to detect and classify pap smears, right, that's used all around the world all the time, then it's sort of a cost benefit. Then it's worth spending, 2 PhD theses to find a really great algorithm to work on this. Not that I worked on that example, I'm just giving you. And I try to use simple and efficient tools whenever possible. If possible, linear. if we can use filtering, then we can use FFT, the power of FFT. Not that every time we used all of these guiding principles, but somehow they seem to have guided a lot of our work along the years, across the years. And then, so the one of the large kind of groups of problems that I looked in this arena over the years is classification. The reason being, if you think about it, classification just assigns labels. That means that lots of problems in medicine, which are really diagnostic problems, are classification problems. You are saying this is normal or abnormal tissue, or this is cancer or no cancer, or you're going to say gradations or cancer or something like that. So classification is a very useful tool. So I'll now guide you sort of through, how are we? We are like 45 minutes on the talk, right? Just tell me when I'm supposed to stop, just to see if I should skip this part or not.
Quarter till is sort of the cutoff date.
Quarter till, okay, it's fine. So let me show you an example of trying to mimic the human. Okay, in a standard classifier, you have generic features and you have a generic classifier. And this is a field that has been studied a lot, and there are lots of features out there that you can use. Haralick features, various texture features, Gabor features, and so on. You have a bunch of, very powerful classifiers. You have SVMs, you have neural networks, you have all kinds of things. But when you want to try to mimic the human, we thought about it this way, that the vocabulary of what we are doing are these features. And then the classification is sort of a grammar that takes these features and puts them together in some way to make a decision. And when we did this, we came up with this histopathology vocabulary methodology. Let's say we have an expert, a pathologist, and that's our collaborator, John Ozelek, and that was my PhD student at the time who graduated a while back, Ramu. And so what John would tell us, I want to describe, let's say, these tissues in a histopathological sample. So he would formulate an initial set of descriptions, and then Ramu has to find a computational way to translate these descriptions so we can implement them on the computer. And then we try to find key terms in these descriptions, which could be color or density or something like that. For example, John would say the bone tissue in an image, and these are histopathological images H&E stained, meaning that there is this pinkish and bluish purplish color to them. So he would say, and this is how the pathologists, this is the most common sort of staining, and all the pathologists in this country spend hours every day looking at these images and diagnosing different things. So he would say bone is pink, has few nuclei, and nuclei are typically dark purple, and they're randomly oriented, okay? So what Ramu saw from this, he's going to take key terms to be R and B values of RGB. R because it's going to do for the pinkish things, and B for the bluish things, and when you combine them, you're going to get things in between. These are two different stains. And so he would say cartilage is blue, has many nuclei, and nuclei are randomly oriented. So many nuclei, so you're going to start thinking, okay, I can define something that's local density of nuclei. And you see what I'm saying. You start trying to extract things that are common in his descriptions that we can implement on the computer to describe these tissues. So now we have to verify, right? So let's say that Ramu says, I'm thinking of bone. This is how bone looks like. And he would say to John, so R is larger than B, so it's more like pink than purple. Local nuclear density is less than 10%. So you see there are not that many nuclei here, not many little blue dots. And the local nuclear orientation has high entropy. So he had to, our collaborator needs to understand these terms, right? So if he understood these terms and says this is describing bone, then this is enough of a description for the computer to be able to recognize bone. If he thinks of cartilage and he starts describing it, and John says, I'm not really sure, then that means that this set of descriptions is not enough for him to recognize. So that was our verification process. And as you go along, eventually we came up with what we call the histopathology vocabulary. Just to show you what it was, so it had background fiber color, and it shows you how this looks like in two different tissues for each one of these. Cytoplasm color, that's the clear thing that exists in the cell. Lumen and clear area density, you see that this one is denser than the fat. Nucleus color, so this one is brighter than that one over there. Density, this one is very dense, that one is less dense, and so on. You see, we came up with 8 of these to describe these tissues. And when we compare this to other features, this is the histopathology vocabulary. These are different standard other features where the combined means the standard features that we have used all combined, and this is the number of features. You see how few of them there are. So clearly, because they were adapted to that problem, I mean, I don't think it's a big surprise that they would work so well, as opposed to 500 random features that, you know, somebody come for generic problems. Okay, so we use the same thing for working on a different different sort of application, but the same types of images, which has actually inflammation of the colon. So we used some of these features and developed more. The idea behind this was that as we would get more, you know, applications, meaning more specific problems, for example, in pathology of the breast screening, we would add more and more specific features to help in describing that particular one, thus building this large vocabulary. Okay, so I'm going to skip this. Another one that we did of the same sort is middle ear infection. So in this is the most prevalent childhood disease of all in the States. And there are 3 diagnostic categories that exist with middle ear. One is normal ear. One is acute otitis media, meaning there is a bacterial infection that makes the membrane of the tympanic membrane in your ear bulge because there is a water buildup and it's infected behind the membrane, pushes out. It's very painful. And there is also otitis media with effusion. That means that there is water that goes behind the membrane. That typically happens when, let's say, you take a shower or you plunge into the pool. The water goes behind. It's not infected, but keeps bulging, pushes the membrane, and hurts. So it's very hard to tell these two apart. So when you take a kid to a pediatrician, and my daughter lived in a pediatrician's office for 6 months with these types of things, and they look with the otoscope into the ear, it's very hard to take these two apart because the kid is tugging on their ear, they're crying, it looks slightly bulged and so on. And what they do is just for sort of precaution, because it could be bacterial, they prescribe antibiotics. So there is a huge overprescription of antibiotics for middle ear infection. And it costs a lot and clearly, you know, reduces the antibacterial resistance. All bad things come with it. And this is how the normal ear looks like. You see the fact that it's kind of blue-gray means that it's transparent. There is nothing behind, so you can kind of almost see through the membrane. The other thing that you can see that it's flat or retracted, while this one you can see that it's pushing outwards, right, towards the outside of the ear. Okay, so for this one, we looked at, they have a number of things they use, the otoscopists, so it means the doctors that I trained just for that. The pediatricians are kind of general purpose doctors. They do this, but they're not very good at diagnosing it. And you can see that there are different things that characterize 3 of the different categories. For example, you can see that color can be white, pale yellow, red, while for normal is gray or pink, right? Very different color, but it's not enough to tell them apart. The other thing that they found out is that the bulging of the tympanic membrane being pushed outwards is the biggest indicator that differentiates between the one that's infected and the one that's not infected. And, okay, there are other indications. And then we found some of our own features. Let's take this apart. So these could be taken as features, right? Color, position, transparency. We can basically describe each one of these computationally. No reason why not. And so that's exactly what we did. We created a set of 8 features that we called otitis media, middle ear infection, vocabulary, okay? Here are the 3 examples of these images. And these 3 basically describe the one with infection. These 2 describe well the normal images, and these 3 describe well the ones without infection, but it's still bulged. And so another thing that we saw, and this was in a medical journal, they had this little like tree, posted of what would, what they found in their. This was extremely useful. So then we said we're going to mimic their grammar as well. So that's what we decided. We devised basically, it's much more complicated to catch the errors in the process because you're going to always make errors. And basically have two stages. In the first one, we detect the one with infection, which is here and here just because I couldn't loop it around in a picture. And in the second stage, we distinguish normal ear from the one with just effusion, meaning just water buildup. This one is not even that important. We didn't even have to do this. But if we want to tell them all apart, this one is important. And just to show you, I'll skip this, how well we did. We had pediatricians, our ground truth is a panel of 7 otoscopists, meaning 7 experts who look at 800 images and rated them, marked them, and they also rated their confidence in marking. So we took a subset of 200 of those where all of them agreed on the diagnosis with high confidence so that we could have a very good set for training. What? They're 2D. They're 2D. Actually, at some point, they were saying that the 3D video would be good, but they found that not to be the case. So the images are enough. And we had 3 general pediatricians do the same. Their success rate compared to the expert otoscopists was close to 80%, right? And this is what we got by doing our algorithm, close to 94%. So we outperform at least general pediatricians. So this would be useful. And we have actually, we have a project with a company that's building an otoscope with our software. And so what you could do is you give it to pediatricians' offices or emergency rooms and so on. And at least it's not allowed to be a diagnostic tool because that would have, we would have to go through FDA. But we can say it's an aid for diagnosis. And so they can see if we say something and this differs from their diagnosis, they can look a few more times. The other thing that we can do, we can take a bunch of images and increase our confidence of doing it right on a bunch of them by simply taking, sort of majority rule. Joy, you had a question.
Yeah, so I'm wondering if you have any data about how your responses and the pediatrician's responses and the truth, I have no idea obviously how that was measured, how they correspond. For example, could it be that the pediatricians were too strict and that they were hesitating to say there's this disease, or is there any pattern that you could have noticed?
No, we asked them the same thing. Remember, the truth is the otoscopies. We don't have ground truth, right? We don't really have the ground truth except it's very hard to know what the ground truth is in this case, right? With the bacterial infection, for example, you could wait and let the kid not have anything. If they have just effusion, it will resolve itself after a little bit of time. The water will kind of sort of seep out eventually. But also if the water stays behind the ear and there is no infection and it's not drained, there is a high probability that it will develop into an infection because it's sitting in an enclosed space behind the membrane. But I don't think that it was anything else that the pediatricians were doing exactly the same thing that the otoscopists were doing. So at least in that sense, it was fair. But again, it's humans saying this is the ground truth. Okay, so this is just to show you, mimicking the human. And now let me kind of go back to the other sort of way we looked at these classification problems in bioimaging. The previous one was mimicking the human. This one was, we were working on, these are images, fluorescence microscope images of different proteins within the cell. How they localize within the cell. So some would localize in some organelles, some would spread out, and so on. And so the question was, because the humans couldn't do it really well, can you tell which protein it is based just on its subcellular localization? And what we did is that we thought that these images, when you make it a large image, look like non-stationary signals. So we wanted to use multi-resolution to help us with classification. And what we said was, let's take a generic classification system which has a feature extraction and classification, a classifier, and let's put, the idea was to say, it is possible that we would see something that distinguishes these classes from each other if we look at them at these multi-resolution subspaces at different scales and resolutions. So to do that, we said, we're going to put a multi-resolution block in front, which means we're going to decompose our image into a bunch of sub-images, all covering one scale and or resolution. And we're going to do classification on each one of these sub-images. And then we're going to get a bunch of results outside. And now I have a bunch of little images and a bunch of little classification results telling me, this one can tell me, I believe it's class A, and the other one can tell me, I believe it's class B. How do we get them together? And so this is what this block does. It's kind of a final arbiter of giving one label out, right? And so to do this, we created this algorithm. So I will use this here, which is multi-resolution, feature extraction, classification, and weighting. Okay. Then we got another problem, and these are Drosophila, fruit fly embryos. And the question that our collaborator had was, there are several developmental stages. This one is how the stomach developed. It's called the ventral furrow. So it's an embryo, and kind of there is a hole that opens up. It starts enlarging, and then finally it closes on top. So there is nothing here. The hole starts opening. The hole has closed, and it's created this like sac inside. Okay, and to see whether developing normally, what they would do, they were trying to identify which gene controls this particular development. So they would turn on and off different genes, and then they would look at the development. And then they know that if this is going normally, this happens at 20 minutes, and this happens roughly at 60 minutes. If they're delayed, this might happen at 40 minutes and this at 80 minutes, or they could be completely abnormal but when the sac never opens or gets stuck somewhere. So they were doing this by hand again. They would take these pictures, then they would look at the pictures and say, okay, at which time this happened, another classification problem, normal, delayed, or abnormal. So we said, okay, why don't we use this algorithm on that application? We did. We found out that there are other things that we need to modify and expand the algorithm. So we kind of went back and forth, getting these new applications. This one is on tissues of histopathological images. Every time, making the algorithm sort of adding features to it. And finally, what we found out, because the multi-resolution block, you can decide whether you want to have basis of frames. For those of you who don't know what they are, they're either redundant or non-redundant representations. And we found out that in all this, and you saw how different this data was, totally different, right? Whenever you introduce multi-resolution, it significantly outperformed. This is about 86%. This is between 92 and 96%. So this is no multi-resolution. This is both multi-resolution. And this makes sense because the no multi-resolution is when the whole thing reduces just to the initial image. So it cannot be worse, possibly, than what it was before. So we found out that this is always the case. Then we found out that the frames, the redundant representations, when we introduce redundancy into the systems, consistently outperform the bases, the non-redundant ones. So that was another one. And finally, this was true across the data set. So we went back and said, okay, I would like to mathematically now understand why this is. There has to be some explanation for this. So we did some work now, very theoretical work, that basically looked at a classification problem and looking at basis versus frames and finding, it was just modeled as a compact convex set, and we could always find a frame set that approximates it arbitrarily well as opposed to basis, which you cannot. And you can think of it as, if I have a circle, the basis is basically a rectangle or a square that you're going to inscribe inside, while the frames are going to be a bunch of these triangles that you can make finer and finer, finally getting you to a circle. So it makes sense. So what I'm saying is about, here what I wanted to point out that at some point we wanted to understand, is there any mathematical reason why this is true, why we are seeing these results? And there is. And so these are all the different classification problems we did in bioimaging that I touched, in one way or another. And then what happened, my colleagues from civil engineering came and said, we have this idea of collecting, monitoring bridges, their health of bridges. Pittsburgh has a huge number of bridges and they're not in great shape. Nobody wants them to fall through. The only way to monitor bridges is to put sensors on or to send inspectors, both of which are costly. So they said, well, there are these cars going on and off the bridge every day. They all have accelerometers inside. Can we get this data somehow, these acceleration signals, and tell something about the health of the bridge by doing that? He said, okay, that sounds super interesting. The first problem that wasn't the biomed arena that I did in this 10 years, but it was super fun. And I said, let me use the same algorithm that I used before to see if I can do that. So we first had simulated data, and it turned out that, yes, we could actually tell these things apart. Now, the problem was, because these are bridges, that we didn't have access to a large number large amount of label data. That's clear, because somebody would have to go and inspect those bridges, right? So we had large number of data points because we were getting it from the cars, but we didn't have them labeled. And this is where the first connection with the graph starts. Because this is the case, we had to move to semi-supervised classification and weighting. Because it's semi-supervised, you have a number of different methods that you can use for semi-supervised learning. One of them is graph methods to use label propagation. And so we started using these graphs and then went back and saw the work that Jose Moura did with my ex-PhD student and then his postdoc on graph signal processing. And we thought, okay, we could use these graph filters to do this label propagation, meaning you have just to think about it, you have a graph and you have just a few labels. You apply this graph filter on it once. What happens, these labels start propagating because it's kind of filtering a value that now is kind of starts smoothing out. You do it again, it will propagate even farther. And so now we went back to this is how the results worked much better. But what I wanted to say is that now we went into the math of actually graph signal processing. And for the last 5 minutes, let me just tell you a little bit about discrete signal processing graphs, which is all I do these days. So this is how we went from biomedical imaging to graphs. So think about it, in standard DSP, you have a set of points, typically in time, and you have a key thing is the shift or the delay, which moves this point into this point. This is your basic filter. You create all filters by taking weighted linear combinations of this, right? So you move, and let's say on finite number of points, they would go back, right? You have a signal. So these are the coefficients now, the values that sit on these time points. And these are the key elements of DSP. And you have a Fourier transform that comes with this. On graphs, you have now points that are, there's complex irregular points, right? The graph shift is now basically the adjacency matrix that tells you how these points are connected. So you see, shift here is this one thing. Shift here is just one entire matrix. So I can now create the signal that sits on these points. I can create filters out of this shift by taking weighted linear combinations of matrices to a power. That's a graph filter. And I can get the graph Fourier transform. Total parallelism, right? So here is how the shift looks. You can assign, this is the filter that goes with it. It's a circulant matrix, as you know, right? For a graph, let's say that I do the same thing. It looks exactly the same, except that I can have it bidirectional, which in this case, I'm going to have a one here as well. Or I can put different values on these links that don't have to necessarily be one. And even if I put, the complex structure for a graph, I still have to index it somehow, so it's going to be a vector in the end. So let's look at the graph signal. Here is a graph signal in time. Here's a graph signal for DSPG. It's really a mapping from the vector space of the graph nodes that are put there in some arbitrary ordering that you want, and you can change the ordering, and then into the space of complex numbers. So basically it gives you a vector, an n-dimensional vector, right? When you look at this tells you that I could use lots of techniques that I have for finite dimensional vectors in signal processing to solve some problems on graphs. If I look at a Fourier transform, so in standard DSP, we know that the circulant matrix is diagonalized by the Fourier matrix, right? This is the Fourier transform. This is the signal back. inverse Fourier transform. And this is the DFT matrix, right? If I look on a graph, I take the adjacency matrix, I do the decomposition, and this is now my graphed Fourier transform, total parallelism, right? I can do exactly the same thing. Okay, and so now, when we looked at this graph, the bridge problem, this actually becomes a signal recovery problem because I'm missing some labels which I want to recover by using on a graph, right? And so what we did is, given a signal with its underlying structure, you want to recover the unmeasured part. So let's say that this is, for example, a graph of cities in in North America, and the graph signal is the temperature. So this is Philadelphia, this is Cleveland. I wanted to put Raleigh, but you were too far away from Pittsburgh. So I need to have 2 cities around Pittsburgh. And so really what I'm asking myself, what's the temperature in Pittsburgh if I don't have the label? And I can do this by using basically the recovery on a graph by, mimicking is a very, very vague word because it doesn't really work as simply as it does in standard signal processing, but you can use the same principles. And so what we did, use this as a mathematical framework. And then, of course, you can expand it, extend it to any sort of a graph. I mean, this was a starting example in this health of bridges. And then we said, well, I can look at this as also as a sampling, saying that those labels I have are sampled out of an original signal, and then I need to recover. So we said, okay, how are we going to do the sampling theory? So this is shameless promotion from the book, for the book that Hamid said. The reason why I'm putting it here, when we were writing a sampling and interpolation chapter, you typically have sampling from continuous to discrete signals, the Shannon sampling theory, but you don't really put it, sort of on paper for all the different combinations we have, right? For infinite continuous to infinite discrete, finite periodic continuous to finite periodic discrete, and so on. And so when we were writing it, we said, okay, let's write how the sampling and interpolation theory would look on each one of these domains. This really was not written anywhere, not that we invented anything new, but it's kind of like a recipe, so why not have it all in one place? The only thing that we didn't do, really, is to do it for the finite discrete, periodic discrete, meaning it's a vector, because we thought it's just too much of the overhead to do it on a simple vector when we can do simpler things. And this actually was a hole in the book that allowed us then to develop sampling theory and graphs. So it's really great. So you can look at it, this is a signal, this is a sampling operator, this is an interpolation operator, you get something back. The question is when x and x prime are the same. You know how to answer this, in the continuous to discrete case, the signal is band limited, you know how to sample to get the signal back perfectly. You can exactly get the same times results, Shannon's theorems, for all the other domains. And so now we had the sampling theory for graphs, where we got, to define the band limited signals, the smooth signals, the sampling theorem, everything as we have in the classical signal processing. But now we can apply to anything. So this was an example of digit classification, classifying images of handwritten digits. And I'm not even sure that's interesting going what the actual data was. This is just to show you what types of problems you may have. So this was basically 10 classes, and these are just the samples that we took for each class from which we could recover the entire thing and classify the digits. Okay, this is the classification accuracy. This just tells you by taking only small number of samples, look, let's say 10 samples, you can already have 75% accuracy. Because we take a small number of samples, we apply the theory to recover the entire signal, and then we classify. As opposed to having 100 samples and classifying. Clearly this is better, but you gain a lot by not having to take all the samples. Okay, so I think, this was just the idea of starting from biological problem or biomedical problem and coming to graphs and show you how we went from one to the other. And I think I'll stop here. Thank you.
We have time for just a couple of questions.
Go ahead.
So what's the typical size of the graph? I guess the real challenge here is the size
Say-
of the graph.
Can you push on this while you're talking?
Say again, what's the size of the graph?
Yeah, what's the typical size of the graph you are thinking here? I guess the real challenge is as the size of the graph becomes large.
I'll repeat.
To find out all the bases, eigenvectors of all this, and different eigenvectors, the full bases. I guess.
What was the last word you said? That you're just asking about the well, the challenge. The question was, what's the size of the graph, right?
Yes.
Well, that's exactly the beauty of this. So we want to be able to deal with very large graphs by taking very few points from them. And instead of having access to all the points or having to take all the points, have very few, assume that this is a sampled signal, sampled graph signal, use our theory to go back to recover the entire signal, and then use that to, classify whatever else you need to do on it. So you can, the point is it can be as large as you want, right? We have, one of them was 10,000 points, another one was 20,000 points. You can do large graphs. We had another example where we had drones flying, following roads, delivering, let's say, packages to disaster-struck areas, right? And if you compute the optimal route for a drone, it will be something. But then if you take the wind into account, the wind would push drone off the road, right? Then you have to compute another route. The problem is to compute these optimal routes is very computationally intensive. So what we would do, we would actually get just a few wind points, and again, use this to reconstruct the entire thing to have access to all the points to compute the optimal route for the drone. So I think the main point of this was that the sampling theory, for example, on graphs, it's very hard to know how to sample on graphs. People would usually put a lot of assumptions, either on the graph, that let's say it's a bipartite graph, or they would assume that they would do this graph signal processing with the Laplacian matrix, which means that the graph is undirected, so it doesn't allow you for the directed graph. And this gives you, it's the same principled way that we have in signal processing, the depth of the sampling theory, and what you can understand from this, you can use it for graphs. So I think that's the beauty of it. At least that's what I'm excited about. Yes?
One more question.
Question. In your bridge problem, what was the edges between the nodes?
What was the
edges in your graph?
It was the similarity.
Similarity of...
So the nodes were individual instances of recorded signals. So basically these were they were like function signals, right? So there was not one coefficient on each node. There was an entire time series recording on that node. And then the edges we computed as similarity. You can figure out how you want similarity. You need to have some sort of a distance between them, and it's up to you how you want to do that, right?
All right, let's thank our speaker again.
Thank you.